So hello everyone, and thank you for those of you who are joining us for today's Malaysian Research Insider session. Welcome back if you've joined us for any of our previous sessions, and a warm welcome to those of you who are joining for the first time. My name is Wen, and I'm a microbiology PhD student and currently based at Imperial College London, and I'll be your host for today's session. So this science chat is co-organized by MBios, the Malaysian Bioscience Scholars, and 100 Scientists of Malaysia, alongside our media partner, Science Media Centre Malaysia. Now I'd like to welcome our speaker for today, Shu Yao, who is currently pursuing a PhD in Cellular Nanoscience. Did I pronounce that correctly? In Germany. Okay. She will be sharing a short presentation to introduce her research area and what she does in the lab. And then we'll follow this up with a Q&A session. So Shu Yao, whenever you're ready, over to you and you can start sharing your screen.
Okay, cool. Okay, hi everyone. Thank you for joining us. And so today I will be talking about this, and I hope we can have some nice discussion about it. So this is the content for what I will be talking about today. So I'll tell you a little bit about how I ended up where I am, and then I will introduce you, give you some background information on the microtubules and microtubule-associated proteins, which are, something I'm really excited and interested in. Then I'll tell you a little bit about the project that I used to do, and then I will finally share what I'm currently doing in the lab that I'm in. Okay, so let's start with education. So I'm actually from Penang, and I went to high school in Convent Light Street, and then I did my A-levels in Taylor's College Subang Jaya in Selangor, and after that I went to Japan, and I did my bachelor's and master's in biological science at Nagoya University. And after I finished my master's, I kind of wanted like a change of scientific environment, so I looked for a position in Germany, and here I am now. I'm actually a fresh PhD student, so I've only been here about 6 months. So I can't tell you too much about the PhD experience other than I've just started, but so far it's been pretty good. That's mostly it about the education, but I just want to talk about Japan and Germany for a bit, because it's not as well known about UK and the US. So I was at Nagoya University, and for the people who know these, you can see here they're like the black and white photo, and there is this old guy. These are actually Nobel Prize laureates that worked at Nagoya University when they had their discovery. So the black and white photo is actually showing the Okazaki pair. Maybe some biologists would know that they are famous for the Okazaki fragments. So they're just very important for one of the big discoveries on how DNA replicates. And then in the middle, you have Osamu Shimamura. He's the guy who actually isolated GFP and used it for imaging. And the last photo is actually pretty fun because it's our auditorium. And if you can notice, the clock tower actually has blue light. And for the longest time, I didn't understand why until we got the Nobel Prize for blue LED. So that was just me flaunting a bit about Nagoya University. So I was actually in the international program, which meant that I actually did not need to learn Japanese to study. So I studied everything in English. And this program, this G30 program, is actually across many universities in Japan. So if you're interested, you can totally apply. But I would say that the Nagoya University has the most extensive program with the most courses offered. So do look it up because it's really great. One thing about Japan is that the education system is very heavy on research. So if you are sure you want to do research, then this is a really good place to go because, for example, by the time I graduated my master's, I had about 3.5 years of research already, and that was full-time research. So you can get a lot of experience that way. Japan is also a kind of affordable, and also the food is really good. So that's another plus for Japan. And these are the other institutes that I would recommend you to look at, which some of them, they have English programs. So you can check them out, yeah. And then after that, I came to university in Germany. And so this building is the building I'm currently working at, and this is the lab that I'm at. Why I actually ended up in Germany instead of like other countries, mainly was because I do basic research. And so Germany is one of the countries who still has a huge support for basic research, and it still gives a lot of funding to it. So I felt safer here. And Germany also has free education, so you don't need to pay for any student fees. And also PhD here is a job, so you get paid, like for sure. There is no entering a PhD here and then you have to find your own funding. Yeah, and I'm also part of the Max Planck Institute. So the Max Planck Association is an association of like research institutes, and they have a lot of money. It's crude to say that, but they have a lot of money, and you need money to study. So, and they're really, really good, and they have very, very advanced research. So to be part of the Max Planck Institute is also a benefit. So you could also check out the Max Planck Institutes for courses, and if you're interested. So now I will talk to you about the science that I'm really interested in. So I'm really interested in microtubules, particularly microtubules in plants. But let's first talk a little bit about what exactly are microtubules. Microtubules are components of the cytoskeleton. And this word is made out of two words, which is cyto and skeleton. Now, cyto means cell, and skeleton is like the skeleton in our body. So it helps to provide support to the cell. As you can see here, in green it's microtubules, and it's helping to keep the form of the cell. So it provides support. But the cytoskeleton is a little bit more, it does a little bit more than our skeleton. It actually also helps to provide like transport of stuff. So it's also a bit like the circulatory system. For example, in neurons, this is really important because the microtubules are what helps to transport like little chemical packages from one end of the neuron to the other. Yeah, so microtubules are very important. So right now we've seen one function of microtubule in special cells, which is in neurons. And you can see that they're very important. And another example of microtubules and their function is in plant cells. So in plant cells, you can see the microtubules here are like around. And this is important because the microtubules are very important for cell wall patterning. So the cell wall is made out of cellulose, and cellulose is made out of a little machine called the cellulose synthase complex. And this machine actually walks on the microtubule in order to make cellulose. And by doing that, the microtubules guide how the cell wall will be patterned. So microtubules are very important here for both transport and support. And another example is in cells that have flagella or cilia. And if you're not sure what a chlamydomonas is, a chlamydomonas is just like a unicellular organism that has flagella. But you have a lot of cells in your body that has cilia as well. And also, guys, you have sperm, and the sperm's tail is actually something like a flagella. And the microtubules are actually inside the flagella, and it helps to give it structure, and it aids movement. So here, microtubules are also very important for the support of the structure, and it supports movement. Now, so far we've talked about microtubules and their functions in special cells. So these are all very, unique cells, and they look very different. But microtubules are actually in every single eukaryotic cell. So all our cells in our body have microtubules. They don't have to be like just neurons or anything. And microtubules are very essential in general cell processes. And one of the most important functions of microtubules is in cell division. So maybe you guys have seen this quintessential cartoon of cell division. So you have animal cells on the left and plant cells on the right. And microtubules actually make up the mitotic spindle. So when I say mitotic spindle, I mean the microtubule machine that helps in mitosis. And mitosis is just a different word for cell division. At least that's what you can see here. And the mitotic spindle is the microtubule's structure here. It's called a spindle because it kind of looks like a spindle shape. At least in most animal cells, it kind of looks like a spindle. So I'm gonna kind of need you to remember these two words, mitosis and mitotic spindle, because I will use it a lot. Yeah, so, okay, moving on. Right, so what exactly are microtubules? Now we've talked about what they're very useful for, but what are microtubules themselves? Microtubules are actually these huge protein structures, and they are made up of little proteins, and these little proteins are called tubulin, and there are two specific types we call alpha tubulin and beta tubulin. And these two little proteins can actually join together to form a dimer, and this dimer is the sort of the smallest unit of a protein, microtubule. These dimers then can arrange end to end to form like a string, a polymer, and this is what we call a protofilament. The tubulin dimers and the polymer, the protofilament, can actually come and join together to form a tube, and this tube we call microtubule. So the construction of a microtubule is what we call polymerization. So you build it and it's polymerized, and depolymerization is the destruction of this microtubule. So one interesting thing maybe you've noticed is the fact that the microtubule actually has one side where the beta tubulin is exposed and the other does not. And the side where the beta tubulin is exposed is called the plus end, or the growing end, because this is where the little tubulin dimers are added to the microtubule so it can be polymerized, so it can be built. And the other end, which is the stabilized end, we just call it the minus end. And this is where the alpha tubulin is exposed in theory, conceptually. And why this is really important is because it gives it directional polarity. So what this means is that, for example, in important cellular processes like mitosis, your things that need to be moved know where to be moved. So let's look at a nice video about mitosis. You can see here in pink is the DNA, and it needs to be separated during mitosis. And in green is the microtubules. And the DNA has to move on the microtubules. First, it needs to go to the middle, and then it needs to separate into two daughter cells. So the chromosome, or the DNA, needs to know in which direction to go. And how this is done is because all the plus ends are actually in the middle of this mitotic spindle, and the minus ends are all on the outside. So what you just have to do is to just move this DNA to the plus end. Yeah. And how exactly this DNA is actually moved is by little transporters. They are just little proteins that can walk on the microtubule while holding something, and that's how they move things in the cell. And since we're talking about transporters, in biology, what we actually, the official name for these transporters are called microtubule-associated proteins, or MAPs. There are many different kinds, and some famous ones are like dyneins and kinesins and a lot of other MAPs. But I'm interested mostly in kinesins, so that's what I'll be talking to you guys about. And the structure of a kinesin is actually a lot like a person, but without the top half of the person. So you have the little legs. And usually these kinesins are they can only walk in one direction, so towards the plus end or towards the minus end. And that's what a conventional kinesin does. But there are some special kinesins, so they're slightly modified. For example, we have kinesin 5 and kinesin 13. You can see here kinesin 5 has basically two of this structure, and they are upside down. And when you have them on microtubules, what this allows is anti-parallel sliding. So because this little kinesin will walk towards the plus end, as it does so, the microtubules will slide past each other. So that's and you can use this inside the cell for several processes. Kinesin 13 is also very interesting because, you know, like the structure, it looks pretty much like the conventional kinesin, but here it's special because the shoes where it attaches to the microtubule is like super sticky, or you can think of it that way. And what that means is that, for example, when the kinesin tries to move, it actually rips out tubulin from the microtubule. And this causes actually the microtubule to destabilize and causes depolymerization. So this kinesin 13 isn't just a transporter, or it doesn't just walk on microtubules, but it can actually destroy microtubules. Yeah, which brings me to what I actually used to study. Now it's going to get a little bit heavy, so good luck. And if you have any questions, just feel free to ask them. Okay, so like I just said, kinesin 13 is actually a microtubule depolymerizer, so it helps to destroy microtubules. And most of this research was actually done in animal cells. In animal cells, kinesin 13 is essential. And what this means is that when you remove kinesin 13, the animal cell will die. And why it's important is because kinesin 13 helps to regulate the length of the mitotic spindle. And so they did some experiments where they reduced the amount of kinesin 13 and found that when you reduce the amount of kinesin 13, the spindle becomes excessively long. And this super long spindle leads to a number of mitotic defects and causes cell death. But the function of kinesin 13 in plants is not very well known. And so that was the aim of my project at that time, was to find out what exactly it does in plants. So the moss, the model organism that I use was actually Fiscomichella patens. It's a kind of moss, and it's here, it's very cute. Why I used it is because it's mostly haploid, and it has high DNA recombination frequency. So this means it's very easy to make mutants for this plant. And also it has developed organ systems. And for example, in this research, I use protonemafilaments, which is in the picture below. And you can see here they're only single cell width, so they're very, very nice to image, and you can have beautiful mitosis imaging. Yeah, so that's the model that I used. And so let's get to the actual results of this research. So the first thing that I wanted to check was whether kinesin 13 is also essential in plants. So I removed kinesin 13 from the plant, at least I tried to, and what we call this kind of mutant is like a kinesin 13 knockout. So we knocked out the gene. And unfortunately, it wasn't essential. So kinesin 13 knockout moss actually survives. And as you can see here, for the most part, it looks a lot like the control. So the normal moss and the knockout moss actually looks very similar. So kinesin 13, at least at first glance, is not essential in moss. Despite that, I wanted to look closer at mitosis because, that was what I was interested in, especially with the huge phenotype in animal kinesin 13 knockdown. So I took some nice videos of mitosis, and you can see here already, for the most part, you don't really notice any huge differences. Mitosis is occurring, mitosis is finishing. But in any case, I needed to do some measurements and understand in greater detail what exactly is happening. So to explain it, it's easier if we look at like snapshots of mitosis occurring. The first phenotype I wanted to analyze was mitotic duration. So how long does it take for a cell to undergo mitosis? And we define it here as the time taken from nuclear envelope breakdown. So when the nucleus collapses and then the DNA is just free until anaphase onset, when the DNA actually separates. And I measured the duration taken, and it was found that kinesin 13 knockout moss actually takes a longer time to finish mitosis. So something is going on, and kinesin 13 is doing something. And when you remove kinesin 13, the cell just has a harder time to complete mitosis. And that's a good sign that kinesin 13 is doing something. So the other thing that we checked was spindle length. And now you remember when you knock down, so when you reduce the amount of kinesin 13 in animal cells, they become really, really long. And already you can see that is really not the case here. But in any case, I measured the length, and what I found is that the mitotic spindle is actually shorter, slightly shorter than the control. So right now, we have found two important phenotypes of mitosis of the kinesin 13 knockout moss, which is it takes longer to finish mitosis and that the mitotic spindle is actually shorter. In order to understand what kinesin 13 is doing, we kind of need to answer this question on how kinesin 13 regulates microtubule dynamics in the mitotic spindle. But microtubule dynamics is actually quite difficult to analyze in the spindle because, as you can see in all those pictures, the microtubules were very, very dense. And so you couldn't analyze a single microtubule to see how the dynamics was changing. So this brings me to the second part of the research, was that I needed to look at microtubule networks that were not so dense. So I could actually see single microtubules. And those microtubules are in interphase. Now, quick refresher, what exactly is interphase? So I talked a lot about mitosis, and that's the cell division phase of the cell cycle. And interphase is basically just all the time that the cell does not spend in mitosis. So basically microtubules that are not in the mitotic spindle. So the first question that we needed to check, the first thing we needed to check was whether or not this kinesin 13 even localizes to the interphase microtubules. And so I checked for this, and you can see here, kinesin 13 is in green, and the microtubules are in magenta. And you can see these little dots on the microtubules, and they're just tracking the microtubules, and you can see that indeed kinesin 13 is here at the interphase microtubules. So this is good. This means that we can use interphase microtubules to analyze microtubule dynamics. Okay, Okay, so then I looked at microtubule dynamics, and first we gotta have our hypothesis. Now, like I said before, in animal cells, we know that kinesin 13 depolymerizes microtubules. And if that is the case, then without something to destroy microtubules, the shrink rate, so the rate at which it depolymerizes, should decrease because there's nothing to cause it to shrink. And the growth rate should increase because, like I said, there's nothing now to cause it to shrink. So first I checked shrink rate. And however, it was found that the shrink rate actually increases. So when you remove a depolymer, at least what we thought was like when you remove a depolymerase, the shrink rate should decrease, but it was the reverse instead. So this hypothesis that we have is actually wrong. But in any case, I went on and I measured growth rate. And again, instead of increasing, the growth rate actually decreases. So this hypothesis is also wrong. So all this hypothesis was based off the fact that we thought that kinesin 13 is a depolymerase, but a lot of phenotypes so far, including the smaller spindle, seems to suggest that maybe kinesin 13 isn't a depolymerase, and it might actually be the reverse. It might be a growth promoter, so it actually, instead of causing destruction to the microtubule, maybe it's actually stabilizing the microtubule or allowing it to grow. But in order to check this, what I did was I took this protein and microtubules out of the cell and did in vitro biochemical analysis. So there are two things that I really wanted to check. I wanted to check if kinesin 13 depolymerizes microtubules, and or if it instead causes stabilization of microtubules, so it allows it to grow better. So first thing I looked at was depolymerization activity. And to do this, what I did was a microtubule depolymerization assay. It's really simple. So you just have some microtubules, you add your protein of interest, and you should see your microtubules disintegrating. And so this is exactly what happens when you use animal kinesin 13. However, when I added mouse kinesin 13, nothing happened to the microtubules. They were perfectly stable, and microtubules will not depolymerize. So at least in the results show that kinesin 13 doesn't seem to have depolymerization activity. So I wanted to now check if it has growth promoting activity. And to do this, I did an in vitro microtubule dynamics reconstitution assay. Very long, very fancy, but what it actually just means is that you recreate the dynamics of microtubules outside the cell. So it's the same. You have some stable microtubules, you add your protein, and now you also add free tubulin, so the monomers, the building blocks of microtubules, so then they can actually take these building blocks and add to the microtubules. This allows it to grow, allows it to shrink, and form dynamic microtubules. So I did the assay, and then I took some nice chymographs, as you can see here. So these pretty pictures here are of what we call chymographs. So chymographs are just time series of images. And here the y-axis is time and the x-axis is length. So you can see microtubules are growing and then shrinking and growing here on these chymographs. To be more specific, like in both the two samples, this is the growth phase of the, at least I've highlighted one part of the growth phase of the microtubule. And how we can measure growth rate is by getting the slope of this line. So I measured growth rate of samples where I didn't have anything and samples where I had MOS kinesin 13. But the addition of MOS kinesin 13 didn't seem to do anything to the microtubules. So at least as far as this study seems to go, MOS kinesin 13 doesn't show any growth promoting activity. But in the course of this essay, I noticed something was happening to catastrophe frequency. Now, what is catastrophe? Catastrophe is actually the transition from growth to shrink. So the specific event in which a microtubule goes from the growing stage to the shrinking stage. In the chymograph, you can see, for example, in the buffer chymograph, you can see that it happens here. And it may be a little bit clearer because of the image quality for the MOS kinesin. When you add MOS kinesin 13, you can see it actually here. And these are catastrophe events. So I measured the frequency of these events and found that when you add kinesin 13, these catastrophe events actually occurs more frequently. So it's possible that kinesin 13 actually has catastrophe-inducing activity. And with that hypothesis, I really wanted to see if that was actually true inside the cell. So I went back to the interface microtubules that we had just now, and I measured catastrophe frequency. Now, let's start with our hypothesis again. If kinesin 13 actually causes catastrophe, then without kinesin 13, the catastrophe frequency should decrease, should be reduced. And I measured this, and indeed, this was the case. When kinesin 13 was removed in the MOS, the catastrophe frequency of microtubules was actually reduced. And so we kind of made a whole circle about what kinesin 13 did, and at least at the microtubule, at least for microtubule dynamics, we think that kinesin 13 actually induces catastrophe. And what this means for mitosis, what this means for the entire plant, is actually a little bit more complicated. So I won't be talking about it here, but if you're really, really interested, you can just ask me, you can send me an email or a message and we can discuss it. But also you could just read it because we already published this work and it's open access so everybody can read it. And this work was done with a lot of thanks to my mentor and also my professor and this other guy who actually helped me a lot with the analysis. And of course, the rest of the lab, for mental, moral support and all that. And also scientific discussions. So that was what I used to do actually. And now I will be talking just a little bit about what I'm currently trying to do. Yeah. So I'm still really, really interested in microtubules in plants. So I changed countries, but I didn't change topics at all. I just really like microtubules. So you remember this nice image of microtubules in plant cell division. And in this case, what I'm interested in now is something called the phragmoplast. The phragmoplast is the cell division machinery in plants. And you can see it in these images. It's that mass of microtubules in the middle. And basically, they are there to help. As you can remember, I told you that microtubules are important for the cell wall formation. A really long time ago, I told you that. So here, what's really important is because you can see in plant cells, you need to build a new cell wall in between the mother cell to form two new cells. And the microtubules are very important for that. Why exactly that is really important is because plant cells can't move around like animal cells. So once you have this cell wall in the middle, that's it. You can't change it and you can't just move around. That's stuck. And if you don't do it properly, that will be a problem. So for example, if you have problems with this, your plants will look like this. So these are some mutants with phragmoplast orientation defects. You can see in the image A, the cells are very beautifully organized, and in B and C, they are very, very badly organized, and this can cause a lot of problems for the overall organism. In these particular pictures, we actually have mutants where POC1 and POC2 are deleted. POC stands for phragmoplast orienting kinesin, so it's also a kinesin, and like its name suggests, it helps to orient the phragmoplast. And POC2 is actually the protein that I'm really interested in. And during cell division, POC2 localizes to the cell division zone. And what I mean by cell division zone is the place where the cell will lay out the cell wall. So in plant cells, at least, this location is actually predetermined long before mitosis even begins. And POC2 will go to this predetermined location and stay there throughout. And we think that and evidence seems to suggest that it's very important for this fragmoplast formation. So somebody actually studied already in vitro what this POC2 does. Now here I wrote POC2 motor domain, and it's a little bit complicated, and I'll talk about it later. But so what basically they studied the POC2, and they found that it can walk on microtubules, in particular, it walks towards the plus ends of microtubules. And with this information, what they did was build a model to better understand. They could understand then how exactly POC2 arranges microtubules in the fragmoplast. So their hypothesis is that POC2 localizes to the edges of the cell. It sits there, and then it captures microtubules that are coming out from the fragmoplast and walks towards the plus end. And by walking towards the plus end, it helps to orient the microtubules in the fragmoplast. And they think that's how it allows for fragmoplast orientation. The only problem with this is that, like I said, we have to talk a little bit about the motor domain. So here is what you recognized before. It's a conventional kinesine. So normally, most kinesines kind of more or less look like this. But POC2 is actually very, very special. It's actually humongous. It is 3 times the size of a normal kinesine. And because it was so big, it was really, really hard to study. So what they did was they just studied the motor domain, so a minimal part. And that motor domain already showed like motility on a microtubule. But like the title suggests here, the motor domain is not enough to understand POC2's function. And one of the main issues with this was because the POC2 motor domain, so this red part, was actually very weak. What this means is that it doesn't have enough energy. It can't generate enough force. And so microtubules coming out of fragmoplast actually is very strong and has a lot of pushing force, and it would not be able to withstand that. And because it won't be able to withstand that, it won't be able to pull microtubules into the correct position. So it's very likely that the other parts of POC2 somehow helps to regulate this force generation, or maybe it helps to, bind this motor domain to other things so that it can actually orient the fragmoplast. And my research now is focusing on these other domains and the entirety of the POC2 protein. And basically I'm done. But I just wanted to say like one few thing, just a small thing about why exactly I'm doing research. I don't really have a huge reason why I'm doing research, because I don't think I'm also super interested in becoming a professor or starting my own lab or anything. I just really like microtubules, and for at least the next few years, I would be really happy to continue studying them, and then we'll see after that what happens. And so I just kind of wanted to say that, because if you also just kind of really like something in science, you can also just go ahead and do it, and then once you're happy and satisfied, you can go on and do other things. Yep. So thank you for sitting through this talk. It was quite long, but thank you. Yep. And you can ask questions now.
Hey, cool. Thanks, Shu Yao. I thought you ended off with some really good advice, actually, because I think so as someone who also works in like basic science, I have to keep reminding myself like I'm doing this because I like doing it. And I don't really know what I'm going to do afterwards, but yeah. So that was very nice. Okay, you can stop sharing your screen now, I think, and then we'll get to some questions, because we got a few questions in the chat. But also, I just wanted to say, and a few of us were just talking about this, like chatting in the background, that you actually explain a lot of those concepts better than my undergraduate lecturers did. So like that was super clear.
Really? I was like so scared.
No, personally, I thought you did like a really good job with your presentation, because like I would imagine like especially people who did like their undergrad degrees in like biochemistry or biology, they probably had to learn about microtubules and cell biology at some point. And I think back then it was just so confusing, but now it's so clear. Okay,
yeah, I'm glad.
Yeah, that's good. Okay, we'll get to some questions then. So I think we'll start off with some general questions about sort of like microtubules, the processes in the cell. So that we have a question here asking about the catastrophe events actually. So what's the biological significance or the function of these catastrophe events?
So it's part of microtubule dynamics, I would say, because microtubule dynamics it's actually a very fine-tuned process. So even like depolymerization, actually the rate of depolymerization can be controlled to how fast and everything. So controlling catastrophe events is also one way to sort of control microtubule dynamics. And in terms of like catastrophe events and depolymerization, why that is important is because when you destroy microtubules, you create the building blocks. And sometimes you just need more building blocks, and the cell actually doesn't continuously make these tubulin. So for example, for mitosis, you actually have to destroy the interface microtubules, and then they will use the tubulin released from the destruction to make the spindle. So it's kind of mainly just for the reorganization, the building, the rebuilding of these many different microtubule structures in the cell.
Okay, so it's almost like it's recycling the
Yeah,
tubulin from somewhere
exactly.
else.
Yeah,
Okay.
it's actually called that.
Oh, okay, cool.
Yeah.
So we had another question asking about kinesin 13.
Mm-hmm.
So actually, is the plant kinesin 13 homologous to the animal one? I assume that's how they've classified them, right? It's because of homology.
Yeah, okay. It is largely because of homology. But so kinesin 13 is particularly interesting because kinesins, how the kinesin family is categorized is as long as they have the motor domain. Mm-hmm. So if they share the homology with the motor domain, they're all part of this huge family of kinesins, and there are like 15. Don't quote me on that. I don't know how many family members there are. I don't remember. There are many. Mm-hmm. And then they're subgrouped into different families based on further homology. But also an interesting thing is that the motor domain actually can sit on the N-terminal, so the start of the protein, or the end of the protein. But kinesin 13 is very, very special because it's actually in the middle of the protein.
Oh, okay.
And why this is interesting, maybe not to anyone but me, but because for the longest time they thought that was the key. If the motor domain's in the middle, then it depolymerizes microtubules. That they thought that was like aha, but that's not the case. Now we're finding more proteins that the motor domain's in the middle, and sometimes they have depolymerization activity, sometimes they don't. So, but historically they have that position.
Okay, yeah, because I was actually going to ask a follow-up question, like something similar to that, about like how many kinesins do plants actually express, and do you know if like someone has then tried to look for the kinesin that depolymerizes those mitotic spindles? Because I assume it's an important process that still needs to happen, right, in plant cells.
Yeah, so I was trying to look for that. That didn't go very well. So I'm not sure if you guys could catch the title of the plant cell paper was actually kinesin 13 and 8. So these are actually the two famous kinesins that seem to show depolymerization activity. And so I actually tried to make like a complete knockout of both kinesins. It did nothing. It didn't do anything at all.
Oh, okay. So neither is essential then for that stage.
At least, so the problem with that was that kinesin 13, there are 3 paralogs. So there are 3, right. And then kinesin 8, there were also 3. So I wanted to speed up the process of making the mutant by using CRISPR-Cas9. Mm-hmm. And instead of, usually what we do is we remove the whole gene by homologous recombination. We give it a template without the gene, and it likes to do that. Moss really likes homologous recombination, and then it just kicks it out. But I wanted to speed this up, so I tried to do like frame shift mutations. Yeah. So I cut a little bit in the beginning, and it frame shift, and I did it to I had the kinesin 13 knockout, and I knocked out the rest of the 8s like that. But it didn't have any phenotype, and I think it's actually because it's not completely knocked out. Okay. Because in theory, it should still have the phenotypes that the kinesin 13 knockout has. It should have at least the kinesin 8 knockout phenotypes, because if it's completely gone, it should have that, but it didn't. So I was really worried that I didn't actually remove them all, and then I needed to leave Japan, so I couldn't follow up on that.
Oh, okay. Yeah, time is another thing, right?
Yeah,
Because
so
actually, yeah, that was someone actually did ask how do you generate knockouts in moss, but I'm guessing in CRISPR-Cas9 that's the one method that you use.
So that's a more newer method, because I think CRISPR-Cas9 was like, what, 2013?
Yeah,
That was when
very
it came
new.
out, something.
Yeah.
Traditionally, moss actually, like I said before, has very high DNA recombination frequency. So what we just did was we just gave it a template, and we removed like the gene part and had the 5' and 3'. And then the moss just like eats that up and then removes the gene. So that was also one of the reasons why moss is very nice as an organism to study, because it's so easy to make mutants. Yeah.
Oh, okay.
Yeah,
So
that's
you really just have to sort of give it the template and it will sort of do its own thing. Oh,
yeah.
That's really easy.
But with Cas9 now, it's actually a lot easier. You don't even need to give it a template. What you do is you just cut the beginning and you cut the end, and the moss will just vomit it out. So
okay.
yeah.
Cool. So we do have a few more other general questions about just motor proteins. So someone asked, like, do you know any diseases, I'm guessing in humans, that are linked to motor protein mutations?
Yeah, so it's very interesting because like microtubules are involved in such essential processes like cell division, transport, and like you saw in neurons. You would think that there would be a lot of diseases, but not as much as I expected. And that's actually because they're so essential that if there's something wrong, the cell just doesn't divide and it dies. Yeah. But some examples of it is actually a lot of neurodegenerative diseases. So you can imagine very easily that if things cannot be transported, then your neurons don't work. So that's a lot of problem. And it actually, one of the, I think ALS, it's related, but it's not the kinesin itself. So what happens is that one of the proteins is called tau. It aggregates and it actually interferes with kinesins to walk on the microtubule. And then because of that, they can't transport things. Yeah.
Right. So because you work on plants, like are there plant diseases that are related to?
To be honest, I'm so into basic research that I never actually thought about what their significance is like outside of like, oh, they just move like this. So it was actually the first time in a very long time that I actually thought about it. So I don't know.
Yeah, okay, that's fair enough.
But yeah.
So someone else actually asked if your research is based more on the biological or physical aspect of such motors.
Good question. So previously it was actually more on the biological aspect. As you can see, most of it was me just trying to answer what exactly this protein does in the cell. But from that, like I still don't know how exactly it does this catastrophe inducing activity, what exactly is happening. And because I really wanted to find out, I really wanted to know how this actually occurs, I really wanted to go into the physical aspect. So that's also part of the reason why I looked for labs that were doing exactly that. And I ended up in Germany because my lab does that now.
Okay.
Yeah.
So going to some more technical questions that relate to like your experiments. So someone asked if you actually have to sync the stage of the cell cycle for your live cell imaging experiments.
Yeah, you usually do that for animal cells because they have very nice drugs that can arrest it at like some specific cell cycle stage. But in moss, you don't. At least you cannot, at least as far as I remember, you cannot do that because these drugs, they don't work on plants because they can't get past and go past the cell wall. But at least for moss, you really don't need that because I think the cells that I was looking at, they had actually quite a high cell cycle turnover time. What do you call that? The cell cycle is actually quite short.
Yeah, okay.
Yeah, and then we also, because we look so much at, we know specific cues, like, oh, when the nucleus looks like this, it's going to divide in 20 minutes. So that was how we did it. We just looked at cells and we looked like, oh, they look like this, going to divide. And it's quite fun because I think the first thing you do when you enter our lab was you spent about 1 month just imaging control moss, just looking for, just like you were basically being machine trained. What do you call that? There's like a word where they like machine learning.
but like with humans doing it.
Yeah, exactly. Yeah, you spend one month doing that, but that was
So
like
you're just like
actually
training
fun.
yourself to identify from the
Yeah.
images. Yeah. Oh, okay.
Yeah.
Oh, that's cool. Um, Someone else asked, how relevant is the polymerization activity of the microtubules to the control of the dynamics of mitosis? So I think they gave like, for example, can I make unequal cell division of a plant cell, or controlling microtubule activity?
Good question. That is a good question.
I think there's actually a drug that can do that, if I'm not mistaken.
I mean, yeah, there is a drug that can cause stabilization of microtubules,
Yeah.
and that's actually a cancer drug. A lot of cancer drugs actually. But in this case of like, for example, to make unequal cell division, at least biologically in cells, that's not actually how unequal cell division occurs. We call that asymmetric cell division. Usually for that, it's the positioning of the spindle. So if you have your whole cell this big, the spindle will be very balanced, but it would be off-centered. And so if it divides off-centered, then this is going to be the division. And that's how the asymmetric cell division occurs.
Right.
But the idea of it, where you can control polymerization activity to make it like unequal, so like one side would be longer than the other, it's pretty interesting. And probably you could somehow do it in vitro. Although I don't know if that's significant. I don't know if there's a significance in cells. Maybe there is.
Because I guess like if you're doing it in a cell, how would you, like if it's a compound or something that affects it, like how would you affect one side but not the other side, right? Because it would diffuse.
Yeah, exactly, yeah. And usually with it's very balanced because it needs like tension, and that's how it detects that like everything is in the middle.
Okay.
So at least in the cell, I don't think I know of any case, although that's really cool. I wonder if there is.
Yeah, it'd be interesting. Yeah, but I can imagine, like what you said, probably in vitro you could potentially,
potentially
a gradient or something of the
I know. Yeah, maybe. But like you said, the main problem would be it just diffusing everywhere.
But yeah, how would you control the gradient of diffusion or something? Okay, someone actually asked, what's the advantage of using plant models to study microtubule dynamics?
Oh, there is, sorry. Like if you notice these, okay, maybe you didn't notice because there were not very nice videos of it, but the moss is like long and you have like microtubules inside. It actually, we always say this, but it resembles a neuron.
Oh, okay.
Yeah, so transport of things inside this long, thin cell is very similar to how things are transported inside the neuron. And because we're people and we care about our neurons. But yeah, so why it's cool, how it's relevant, maybe like that, but maybe not at all. But that's also the thing about basic research. A lot of times there is not an immediate application for it, but it's just really cool.
You never know, like, right, 10 years down the line or someone's going to be like, actually this is going to be really useful for this random application.
Yeah, exactly. So we just put it there in case it's useful in the future.
Yeah. So I think someone in the chat just mentioned that they actually gave plant pathogens and microtubules a quick Google, and they found that there are pathogen effectors which can target microtubules to cause virulence. So they put that link there if anyone's interested. And then I think we're going to move on to some sort of like future questions and also like questions about like education. So someone asked, like you mentioned at the beginning, that Germany supports basic research, which is great. So what's the difference between basic research and sort of other kinds of, I guess, maybe more application-based research?
Yeah, I mean, we just talked a little bit about that, right? So basic research is really like fundamental research. If you want to look about, if we just take microtubules and kinesins as an example, basic research will be just like, so how is a microtubule built? How does this tubulin bind to this tubulin? Why does it do that? And then like a huge field of it is just like taking microtubules and analyzing like, does it bend this way? Does it bend the other way? Or because they are made from protofilaments as well, they're microtubules with different sizes. So they can be really fat, really thin, and it's really just the extent of what we know, and that's basic research. But if we talk about other kinds of research, or we would usually, I think what that would refer to is like transitional research. If in microtubules, then it would be definitely about like diseases. And microtubules have a huge place in cancer because because it's so essential in mitosis. And the idea is that cancerous cells are very, very mitotic. They love to divide. And normal cells don't really divide that much. So if you inhibit this division, chances are you'll be targeting cancer cells and not normal cells. And a lot of the drugs out there is to target exactly this. So we mentioned a little bit just now of Taxol. So Taxol actually binds to microtubules and causes them to stabilize, and then it can't divide, and that will kill the cancer cell. Yeah, so that's a little bit of the difference. And like sometimes I do think about like, it would be nice to, actually see a point in my research, but I think I cannot do that. I will be too stressed out. Yeah.
Oh, but I feel you. Yeah, as someone who's also doing basic research, yeah, I totally feel you about that whole. Like, I wish like people could see what the point was.
Yeah, because like sometimes I don't see the point either.
You have to remind yourself over and over again. Okay, the last question for future questions, you kind of touched on a little bit already at the end of your talk, but it's basically about like what you plan to do after your PhD.
So like, to be honest, I do have some kind of plan, but it's very flexible. It was the plan I made at the time, knowing everything that I knew. So my plan was that I like science and it's okay so far, and I will just keep doing it until it's not okay anymore. And but because that's also the idea that I have, so as I'm on it, I do have sort of things that I'm doing in case I don't want to do it anymore. So for example, I'm trying very hard to learn German properly, because chances are if I don't like academia, then I'll have to look for a job. And if I plan to stay in Germany, then I would need German to do that. But also I really like science, so I was thinking that maybe one possible job I could do was in scientific publishing. So I also try to go and attend courses and just get more exposed. So this is the kind of like the time to do it. Yeah. But
I think that is something that a lot of people are maybe not too aware about, but having a PhD doesn't mean you only have to be a researcher. There are also like opportunities in, like you said, like publishing. So you can work for the journals as like part of their editing team, where you read all the papers and you decide whether they get to go in your journal. Yeah. So yeah, I think that's all the questions that we have. This was a really great session. Like, I learned a lot. So thank you so much for sharing on your work. And of course, thank you as well to the audience for joining us and for asking those really great questions. So next Saturday, we will be joined by Jia Zheng, who is a recent master's graduate from the University of Heidelberg, so also in Germany. He's an incoming research assistant at the Whitehead Institute in MIT, and he will be sharing about regeneration in planarian flatworms. So if anyone is interested in stem cells, developmental biology, don't miss out on that session. For more updates on future Research Insider sessions, like the MBios and 100 Scientists of Malaysia Facebook pages, and we'll see you guys next Saturday. Thank you, Shu Yao.
Thank you.